No confusions. We are discussing about ETC, electron transport system. Electron transport system, the site is, it occurs between the mitochondrial matrix and membrane, inner mitochondrial membrane. So this membrane is having the enzymes. Five enzyme complexes are embedded in this membrane, as well as two electron carriers are embedded. What are the five enzyme complexes name? The first one, it is NADH dehydrogenase. The another is FADH reductase or succinate reductase. Third is cytochrome BC complex. Fourth is cytochrome oxidase. And the fifth enzyme is ATP synthetase. Got it? Next point is the between these two electron mobile carriers are present. That is ubiquinone is one and cytochrome C is the another. So ubiquinone will be carrying the electrons which were released during the during the oxidation of NADH and FADH will be carried by the ubiquinone and transferred to cytochrome BC1. That is the third enzyme complex, right? So during this reduction process, H plus ions were released into IMM, that is inner mitochondrial membrane. Then the electrons which were released, which were collected by the cytochrome BC1, were again received by the carrier, that is cytochrome C, will give that to cytochrome C oxidase enzyme complex, which is the fourth one of this ETC. Fourth one of this ETC, right? So this enzyme complex, in fact, transports the electron to ATP synthetase, the last enzyme complex, as well as it intakes the H plus ions. It intakes the H plus ions which were there in the IMM. Why these H plus ions are entering inside through this cytochrome C oxidase? Why not between the other membrane, other enzyme complexes? Simple logic is cytochrome C oxidase has the permeability for H plus ions. The remaining enzymes are impermeable for H plus ions. So only H plus ions can enter, again came back to mitochondrial matrix through this enzyme one, through this enzyme complex only. So while entering, these H plus ions are utilized for the for the manufacturing of a water molecule. Through, like, at the end, those electrons which were transported are utilized by ATP synthetase in the binding of inorganic phosphate to ADP, that is adenosine diphosphate. Finally ATP molecule is produced. Coming to the calculations part, as I discussed earlier, one NADH molecule, one NADH molecule reduction, sorry, one NADH molecule's oxidation, removal of H plus ions, the H plus ions removal releases 4 H plus ions. Okay, so sorry, one NADH molecule increases H plus ions here. The electrons which were released by one NADH molecule are able to produce 3 ATP molecules. So one FADH molecule reduction, sorry, one FADH molecule's oxidation is able to produce 2 ATP molecules here. Meaning the number of ATP molecule produced at the last step is dependent upon the electrons which were released during the dehydrogenation of this NADH or FADH molecules, right? So this is all about ETC and oxidative phosphorylation. As I mentioned earlier, ETC is clearly restricted only this till here, the transporting of H plus ions between the transporting of H plus ions between the enzymes. Oxidative phosphorylation, OP, this step. So ideally, by the oxidation of all these oxidation of NADH and FADH molecule, phosphorylation is happening to produce ATP, right? So that's why this step is named as oxidative phosphorylation. So let's quickly have a glance at the video, and we'll continue the discussions, sorry, doubt solving sessions here. So you can see here NADH molecules are binding and converting back to FAD plus and NAD. So that is transporting the electrons between those systems, and those electrons are utilized to produce a water molecule, and finally those H plus ions are entering, and ATP molecules are produced back at ATP synthesis. So the free oxygen molecule present in the mitochondrial matrix are binding at the fourth complex, you can see, are binding with the H plus ions, finally producing a water molecule. And ADP is also converting into ATP. So NADH and FADH2 molecules. Clear, I think. So this is the process.